Thank you for visiting the website of the Dyslexia Association of India. The DAI is a registered charitable trust which works on an NGO and a not-for-profit platform. The main thrust of the association is to create awareness of dyslexia and other learning disabilities like autism, ADHD, dyspraxia, dysgraphia, to name a few. We work on assessing children, identifying where the cognitive problems rest, and helping children with neuroplasticity. There is a great deal of uncertainty amongst the parents in India as to what dyslexia is all about. Although there has been extensive research on dyslexia, there still exists a great deal of confusion in India about what dyslexia actually is. Over millions of years, evolution has ensured that the human brain has evolved in a manner that its functions have gradually got lateralized. This lateralization of the human brain is what makes us unique and has allowed us to achieve the degree of complexity that we see around us. While our brains have evolved to grasp phonological sounds that make up speech, and this specialization is evident when children learn to correlate the sound to structure of a specific language. The human brain was not created to learn and to read and write. Reading and writing have to be learned, and to read and write, we need to use three very specific areas of our brain which lie within the left hemisphere of the human brain. In the front of the left hemisphere, between the frontal and the temporal regions of the brain, there is an area for sounding out words which identifies the phonemes in a word when it is heard, like the word I in India. Similarly, in the posterior and between the occipital and parietal areas, on the left side, there is an area at the base of the brain which takes the word and pulls the word apart even more by analyzing the syllables and phonemes, as in India. The third area, which is also known sometimes as the visual word form area, is responsible for detecting word forms and allowing us to instantly recognize the word we are reading without having to sound them out repeatedly in our head, or to keep attempting to break down the words being read into their different subdivisions. The more the activation in this area, the better a child can glide through print without hesitation and any breaks. Research now indicates that if this area is affected and does not function smoothly, it can impact our ability to read, and this area is supposed to function differently in some children who are assessed to have a specific learning disorder or dyslexia. Children with dyslexia have a difficult time trying to gain access to the region of the brain which holds the word analyzer and the word detector. This neurological block can result in these children attempting to compensate and rely more heavily on sounding out words, and they may even try to compensate by using areas from the right hemisphere from where they attempt to generate visual clues to decipher words. In fact, when a child is assessed to be dyslexic, the primary deficit that we see is one of phonological origin, where the child is not able to process phonemes accurately and stumbles on phonological processing and decoding. These children can have very good visual spatial abilities, and this compensatory strategy, where they rely on visual clues to decipher word form, can result in a roadblock in academic achievement the moment they progress to higher classes, when learning to read becomes reading to learn, and parents find that an otherwise seemingly bright child begins to slide and slip through the educational net. Unlike normal cells in the human body, which replicate or divide for growth, neurons in the human brain are programmed to migrate in a predetermined manner, and this migration is a fundamental process that determines the final allocation of neurons in the nervous system, establishing the basis for the subsequent wiring of neural circuitry. From cell polarization to target identification, neuronal migration integrates multiple cellular and molecular events that enable neuronal precursors to move across the brain to reach their final destination. The correct positioning of neurons during development, achieved through direct migration, is the basis for proper brain function. Several decades of research have yielded a comprehensive map illustrating the temporal and spatial events underlying neurogenesis and neuronal migration during development. When we see impairments in attention, hyperactivity, comprehension of text, mathematical difficulties, short-term memory, sequencing of letters, word sounds and motor acts, eye movements, poor balance, and general clumsiness in our children, most of us think that these are normal childhood issues and that children will grow out of these problems with time. Research now shows us that for children who develop learning disabilities, neurodevelopmental abnormalities when neuronal migration either stops or goes awry leads to differences throughout the reading networks in the brain. The deficit in neuronal integration, where they do not become a part of the information processing networks, may prevent crucial neurons not developing neuronal processes like axons and dendrites that allow them to communicate with other neurons. Connections called synapses are formed with the target cell. Synapses allow for the transmission of electrochemical information, which is the essential means of communication in the brain. In children who later show learning disorders of different etiologies, what remains common is that the formation of precise connections between developing neurons and their targets has not occurred. After the initial exuberant growth in terms of both the number of connections formed and their topography, that supports different aspects of the process of emergent connectivity in the developing brain. Pruning can be observed within major brain areas and pathways with synaptic plateaus which slowly decline in pathways as diverse as the corpus callosum, the thalamocortical pathways, the corticospinal tract, and pathways linking the temporal lobe and the limbic system. The modulation or the stabilization or elimination of pathways and pruning is so rapid that as axons seek their targets, they very rapidly sample the surrounding space, forming and retracting synaptic connections in a dynamic manner. Although we are beginning to understand the biological foundations of reading and writing disorders, it is important to recognize that there is a great deal that remains unclear. Although there may be a genetic predisposition to factors that contribute to reading disorders, clearly there is much more than a one-on-one correspondence, as reading skills in general are related to both visual and auditory processing speed, with reading skills occurring on a continuum and involving multiple processing systems. A crucial issue that we need to understand is that if your child is showing developmental delays and cognitive indicators where the observable academic and social output does not match the perceived intelligence of the child, it is a red flag and should not be ignored by the parents. The opportunity cost of an assessment in terms of time and resources that a parent spends when a child is small is far less than the potential loss of academic and social markers of long-term success that would remain for the rest of the child's life if left unidentified.
We would now like you to meet a few children with exceptionally high IQ who have been assessed with having dyslexia.
When I get my mark sheet, I feel very sad that my friends are getting very high marks and I'm getting just 4.7 like that out of 20. My teacher said that you don't study and you are just having a fun in the school. You are just wasting your money, parents' money. When my mother also asked that how was your paper, I just don't want to tell her that my marks is this, because they were also very sad that in school I was just having a blank mind. My whole body will like wish that feeling I get in the school, in the examination hall.
रास्ते में भूल जाती हूँ। And I feel very bad. I always want to cry, but I told my friend that I always want to cry. she always hit me on my head, and I feel very sad. So I feel very scared. I feel very scared that if I will, she will hit me if the spelling will be wrong.